Hey everyone, and welcome to Spring 2016, our very first chapter on blood. Without further ado, we'll go ahead and get started. These page numbers correspond with the orange textbook, so if you have the blue textbook, the newer version of it, these page numbers won't be quite right, but we're going to be in the blood chapter, and the pages that you're omitting are primarily those pages that are concerned with blood typing, because that is an activity that we completed in lab, and for us, we did a virtual blood typing activity. All right, so jobs of the blood. Blood really has one really basic function. Everything else on this list is kind of like a sub-duty of it. But the transportation of dissolved substances is really the major function of blood. One of the most important dissolved substances that is transported by the blood via the hemoglobin is oxygen. Oxygen and carbon dioxide carried to and from the lungs is a very important aspect of the blood itself. However, that's just the job of the red blood cell primarily. However, the regulation of pH and ions, restriction of fluid loss at injury sites, defense against toxins and pathogens, that's the job of white blood cells, stabilization of body temperature with the fact that blood circulates throughout your body, so along with that circulation, it can distribute that core temperature, that body temperature. All of these are other jobs by other aspects or components of the blood itself. And what we're going to see is that blood isn't just made up of red blood cells, which we talked about already in our virtual lab experience, is that the blood is actually made up of a couple of different elements. You have your formed elements, and then you have your liquidy portion of it that we call your plasma. So looking at the composition of blood, when you cut yourself, primarily what's always coming out is whole blood. Very rarely do you have just plasma eking out, although that can happen. If you cut yourself, you have whole blood. And what whole blood consists of is, it's red, it's mostly red blood cells. So red blood cells are the primary constituent of your whole blood. In fact, 99.9% of your whole blood are, for the formed elements, are red blood cells. White blood cells count for less than 0.1%, and platelets count for less than 0.1% as well. Now, the other portion of your blood, now the reason that it's showing it this way, where the plasma is on top and the formed elements are on the bottom, remember we talked about centrifuging, or putting the blood in the tube and sticking it in a machine that spins around and round and round in a circle, and the heavier elements go to the bottom? Well, these red blood cells, white blood cells, and platelets, or these formed elements, if you will, they're much heavier, so they're going to sink down to the bottom. The more liquidy portion of the blood is going to be the plasma. So primarily the plasma consists of water. In fact, 92% of your plasma is just water. So it makes sense why overhydration or underhydration would affect your hematocrit level. And remember from lab that hematocrit is what measures the packed volume or packed percentage of red blood cells. Other solids on there, we have different types of ions that you find: potassium, sodium, chloride ions that are also going to be traveling in the blood. About 1% of that is the plasma. And then the plasma proteins, 7%. And there are various different plasma proteins that we're going to become familiar with. So the clinical note. Now, since we have a little bit of an understanding on what the role is of plasma and the job of those red blood cells and those formed elements, we can extrapolate that information and exploit it to use it for the good of mankind, if you will. Plasma expanders are defined as a temporary increase in blood volume. Now, it's a temporary increase in blood volume because what will happen is that whatever we've used to expand the plasma is going to leak out into the lymphatic vessels, and then may even leak out into the interstitial tissues. And if you'll remember, interstitial tissues are just kind of all the goo that's surrounding all of your other tissues. Blood is, if you'll remember from ANP1, is a type of connective tissue. It's one of two fluid connective tissues, and that kind of makes it specialized. Lymphatic lymph is the other type of connective tissue, fluid connective tissue. So different types of plasma expanders we have are isotonic electrolyte solutions, like a saline solution. This is probably of the list the most short-lived because it very easily will leak into the lymphatic vessels. so you don't really get that increase in volume. One of the reasons why you would want to increase your blood volume temporarily is that if you've had catastrophic blood loss, and your body takes about 10 days to replace red blood cells, so you're not going to be able to replace those red blood cells immediately. But if your blood pressure, if your blood volume goes down, your blood pressure is going to go down as well. And if your blood pressure falls down too much, you can be in what we call crisis, which means that your organ systems will start to fail. Your heart will stop pumping, your lungs will stop taking in oxygen, your kidneys will stop doing their thing. So it can lead to organ failure, which can lead to death. So if we need to get your blood pressure back up, and we see that it's the fact that your volume is low, because the two are related to one another, your body will normally do that under hormonal control. It will actually allow your kidney to release erythropoietin in order to make more red blood cells, and it'll also allow your kidneys to release renin that will then trigger angiotensin II, that will retain more water and trigger the antidiuretic hormone. So there are hormones, if you remember from the very end of AMP1 that we talked about, that can also increase your blood volume. But if we are in a crisis situation, we can't artificially stimulate those types of conditions. Ringer solutions contain potassium chloride and calcium chloride ions, and they can slow down that rate of diffusion of plasma going into the interstitial tissues or into the lymphatic vessels. Isotonic saline with purified human albumin is another example of an expander. It'll also slow it down a bit because now we have this plasma protein that keeps the blood, or keeps the plasma from leaching out too quickly. And then large amounts of carbohydrates will also do that. But eventually, through phagocytosis from your various different cells, those carbohydrates will be consumed and removed. So keep in mind that this is just a temporary solution to a problem of low blood volume, which relates to low blood pressure. Expanders don't increase the amount of oxygen carried by the blood, only the volume. Why is that? Well, if you listen to the preview lecture of this, then you probably already understand why, or just from listening to our discussion from our lab and also from today, is that we're just increasing the plasma. We're expanding the plasma. We're not doing anything with those red blood cells. It's the red blood cells' job to carry the oxygen. So you can put as much plasma in there as you want to, but it's not going to increase the oxygen carrying capacity of the blood. So of those formed elements, there are three of them that you should know. All three of them that we've looked at already in lab. Erythrocytes, or red blood cells, job is to transport oxygen. Number two, white blood cells, or leukocytes. They're part of your immune system, so they have a role in defense. And then you have your platelets, which have a role in clotting, and we'll talk about them more at the end of the semester, or at the end of this chapter. So those are cell fragments. The physical characteristics of blood. It's about 100.4 degrees Fahrenheit. It's the normal temperature of it. So remember, the job of the blood was to regulate body temperature. It circulates through all of your extremities. If your extremities were to get cut off from blood circulation, not only would they be deprived of oxygen and those tissues could die, but you'll probably notice that it starts to get cold because you're not getting that temperature distributed throughout your body. It's very viscous, which means that it's not as runny as water is. It's more like a maple syrup or a molasses sort of thing, very thick and viscous. And it's slightly alkaline. Alkaline is anything with a pH above 7. So the pH of blood is between 7.35 and 7.45. I want you to commit this pH to memory, not only for this chapter and this set of notes test, but also throughout the remainder of the semester, because we're going to come back to this discussion when we get into the urinary system. What's in our plasma? Primarily water. Like 94% of your plasma is water. And if you didn't remember that, we'll go back. I'm sorry, 92% of your plasma is water. I said 94%. So 92% of your plasma is water. So it's primarily consisted of water. But for these plasma proteins on here, three types of plasma proteins I want you to know. We have albumins, globulins, and fibrinogen. The job of albumins are to transport substances such as hormones and fatty acids. At the end of AMP1, we talked about how we can have free hormones, or we can have those hormones that are bound to plasma proteins. And those hormones bound to plasma proteins have a longer lasting effect. Where here is that plasma protein that we talked about. We didn't give it a name so much in AMP1. We just said that it's a plasma protein. This is that plasma protein, albumin. So it transports substances throughout your body, and it has an attachment to hormones such as thyroid hormones and steroid hormones. Globulin proteins, also known as antibodies, and they can also be called immunoglobulins. Now, we talked about antibodies in our virtual lab experience, that you have type A blood and you have type B antibodies. If you have type B blood, you have type A antibodies, or immunoglobulins. They transport small hormone-binding proteins, allolipoproteins, and steroid-binding proteins. It's kind of a secondary job of the globulins. I'm more concerned with you understanding that the globulins primarily are your antibodies. And these antibodies are not just for type A blood and type B blood, but these antibodies are also important for fighting off infections. And we'll have a much more in-depth discussion about this when we get into the lymphatic system. And then finally, fibrinogen. Its job is to produce fibrinogen can be converted to fibrin through a cascade of chemical events that happens in your circulatory system if you are to get cut or to bleed or to have some blood loss. So fibrinogen has a role in the clotting process. So it produces these long and soluble strands of fibrin after it's been converted through the cascade of chemical events that happens in the clotting. The liquid part of the blood sample is what we call the serum. The serum in which dissolved fibrinogen is converted to solid fibrin, that's where that conversion takes place. But keep in mind that fibrinogen usually doesn't spontaneously turn into fibrin. There's a whole cascade of chemical events in which allows that to take place. However, the fibrinogen being converted to fibrin happens in the liquidy portion of the blood sample, or in the fibrin. The serum is involved in blood clotting, and we just explained why that is, because fibrinogen is in the serum and fibrinogen is converted into fibrin, but it also helps with removing calcium and other materials. So other plasma proteins that we would find, we have peptide hormones that are found circulating in the blood, insulin, prolactin, glycoproteins, thyroid-stimulating hormone, follicle-stimulating hormone, luteinizing hormone. Those are all other entities that we can find in the plasma. So now on to red blood cells, also known as erythrocytes. They make up about 99.9% of your blood-formed elements, and remember the other less than 0.1% are your white blood cells, and then you have your platelets there. Hemoglobin is the main constituent of red blood cells. They don't have a nucleus, as we saw in lab. Those flat donut-shaped pinkish kinds of cells don't have a nuclei. They don't really need one. They lose it in part of their maturation process. Hemoglobin, super, super important. So important, in fact, I'm going to put a little sunshine by it. Hemoglobin is responsible for carrying oxygen to your cells and carbon dioxide to your lungs in order for it to be exhaled. So that primary job of the red blood cells of transporting dissolved gases is directly as a result of hemoglobin. Because of hemoglobin, it has this iron or heme group in the middle of it. It gives blood that red pigmentation. Hematocrit is one of the blood indices that we talked about in lab. It's how we measure our packed cell volume, or the percentage of red blood cells. And keep in mind that hematocrit by itself is not going to tell you a whole lot about your patient, because there are other indices that we need to consult with as well, including your mean cell volume, which is the average volume of your red blood cells, as well as the average content of your hemoglobin. It does give you a little preliminary information because there are certain conditions that can affect your hematocrit. Dehydration is going to increase it, so it's going to look like you have more red blood cells, a higher volume of red blood cells than you actually do. And the reason it looks like you have higher volume is because you have less liquid part, we have less water because you're dehydrated. Internal bleeding or red blood cell formation problems can decrease it. Now remember, after you have a major hemorrhaging episode, immediately following, if you were to measure hematocrit there, that your hematocrit is not going to drop significantly, because when you hemorrhage, you lose out whole blood. You lose the plasma as well as losing the red blood cells. However, over time, because it takes about 10 to 11 days to make more red blood cells, what we'll find is that we will see a drastic decrease in hematocrit. Or if you have problems with your hormone erythropoietin or any of the other hormones or entities that are responsible for making red blood cells, if you have a problem forming red blood cells, that can also decrease it as well. Sickle cell anemia would decrease hematocrit. So the structure of red blood cells are much thicker on the outside and thinner in the middle, and that's why they look more donut-shaped. I'll attempt to try to draw that. Bear with me. They kind of look a little something like this. They have a disc shape to them. Kind of something like that. Kind of looks a little bit like that, where it's thicker around the edges than it is in the center of it. And that's, again, just because of a lot of the cytoplasm and fluid and hemoglobin is found on the edges of it. So it looks like you can see straight through it, but really it's just very thin. If we look at red blood cells in a capillary section here, which is what we're doing at, we find that they form these little stacks called rouleaux. And because of the shape of red blood cells, because they kind of have that donut shape to them, they're very flexible. They can fold over on themselves and not lose their integrity, not lose their ability to transport gases and oxygen just by folding over. Now, they completely, the hemoglobin completely loses its shape. That's another point to consider. But because they can fold over and they're thinner in the middle than they are on the outside, they can flow through very, very teeny tiny capillaries that are microscopic, in fact, and they form these nifty little stacks that we call rouleaux. Because red blood cells don't have a nucleus, they can't divide on their own. They did have a nucleus at one point in time, but it got kicked out. So they don't have a nucleus, they can't divide on their own. They also don't have any mitochondria, so they can't make any energy, and they don't have any ribosomes. So they lack nuclei, which will give instructions for making new red blood cells immediately, like a red blood cell just divides and then makes another red blood cell. They don't have mitochondria, so they don't have anything to maintain its energy source or to continue to make energy, and I'll tell you why they don't have mitochondria in a second. And they don't have any of these ribosomes. So these ribosomes are responsible for making proteins for protein synthesis, which is an energetically expensive activity. So the lifespan of red blood cells is not very long. There's really no means for repair for those red blood cells, and they have to rely on anaerobic metabolism, so they only live for about 120 days. It only takes you about 10 days to make new red blood cells, but they're not going to be this indefinitely long-lived cell. Now, if you remember from an introductory biology class or even from AMP1 or a microbiology class, any other science class, what is the role of a mitochondria? The mitochondria is the powerhouse of the cell. Excellent. And the mitochondria is the powerhouse of the cell because it made energy in the form of ATP by using what gas? Oxygen. Absolutely right. So if a red blood cell had mitochondria and a red blood cell's job is to carry oxygen, what do you think would happen if they were both present? What would happen to that oxygen that's supposed to be carried? Absolutely right. It would be used up by the mitochondria. So it's a very exquisite design that they don't have any mitochondria. Job of hemoglobin is to carry oxygen. Very important. We've said that several different times. Anytime your professor says something more than once, not just in this class, but in any other class, that probably means it's a good idea to write it down because it's probably pretty important. So the job of hemoglobin is to carry oxygen. In the peripheral capillaries, or the capillaries that are furthest away from the heart, keep in mind those biological directional terms that we use, that if you're medial, you're closer to the center line, or we would consider that more proximal to the center line. The center line, you can kind of just take a line and go straight down your heart, down to your belly button, all of that's the center line. So anything close to that would be more proximal. Anything further away is more distal. So the further away you get from your heart, the further away you get from that center midline, the lower the oxygen concentration in those capillaries. So in those, on the periphery or on the fringes of your circulatory system, you have these capillaries. So the oxygen concentration is going to be very low. The way that hemoglobin works is that hemoglobin will be triggered to release oxygen when the ambient oxygen, or the oxygen that's around in that area, is very low. Conversely, hemoglobin will bind to carbon dioxide when it's very highly concentrated, and then bring it back to the lungs. And we call that carbon dioxide and hemoglobin carbaminohemoglobin. So I'm a red blood cell, excuse me. I'm a red blood cell, and I'm leaving the heart, and I'm going down to the pinky toe, and down at that pinky toe, there's very little oxygen, so I drop off my oxygen, and at the same time, since there's little oxygen, there's probably lots of carbon dioxide, because that's the byproduct of cellular respiration, and those cells have definitely been cellular respiring. I pick up that concentration, I pick up that carbon dioxide, then bring it back to the heart and to the lungs. Another important aspect, of hemoglobin is that it has iron ions in it. That iron associates easily with oxygen. In fact, it has a very high affinity or like for oxygen. We call that oxyhemoglobin. And when oxygen is attached to that iron ion in the hemoglobin, then it's going to give it a very bright red color. So it gives a nice bright red color to it. However, these iron ions can dissociate easily from oxygen, which is a good thing because we want this oxygen to be able to be dropped off in the cells. So the oxyhemoglobin, which means that this is hemoglobin that has lost its oxygen, has more of a darker red color. So bright red blood means that it's highly oxygenated. Darker red blood means that the oxygen is much lower. Now that's not a bad thing because it could be the case that this blood has just dropped off its oxygen and it's making its way up into a vein via a vein to come back to the heart to pick up more oxygen. Bright red oxygen just means that it hasn't dropped off its oxygen yet. So here is an artist's rendition of a hemoglobin molecule. Hemoglobin is actually 4 different chains. It's a protein, so keep in mind hemoglobin is a protein that carries gases. It has 4 chains, 2 alpha chains and 2 beta chains. So there's alpha 1 chain, there's alpha 2 chain, there's beta 2 chain, and there's beta 1 chain. And in the center of each of these, you have this hemi group. There's a hemi, there's a hemi, there's a hemi. And in the center of that hemi group, we have that iron, which has the ability to bind to oxygen. Fetal hemoglobin is a little bit different than what we consider adult hemoglobin. And when we mean by adult, that just means that you're not a fetus anymore. So you can be 3 weeks old and still have adult hemoglobin. Fetal hemoglobin, if you are carrying a fetus, has a very strong form of the hemoglobin that we find in embryos and fetus. It's able to it has much higher affinity for oxygen than even your hemoglobin does. So what it can do is that some of mom's blood circulates into her uterus via her placenta, and the baby is connected directly to the placenta. It's got 3 blood vessels in the umbilical cord that connect it to the placenta. So when the blood comes in from mom into the placenta, baby needs to get oxygen that way. Your baby doesn't breathe air. So it's not getting any oxygen that way. It's in a very aqueous, watery solution, not using its lungs to breathe. Towards the end of the pregnancy, your baby will start to play around and practice using its lungs. If you've ever had a late pregnancy ultrasound, you can see, or the doctor will look and say, yep, your baby's practicing using their lungs, or you can see them taking inhalations in and exhalations out, although it's just amniotic fluid that's going in and out of their lungs. Anywho, they need to get oxygen. They need to steal it from their mom. So their fetal hemoglobin, those red blood cells have a super high affinity for oxygen, and they can pull that oxygen very easily. And what we found is that if we can turn adult hemoglobin into fetal hemoglobin, then it can be used to treat sickle cell anemia or thalassemia, which means that you have there's an issue with your platelets. So to treat sickle cell anemia, for people that have sickle cell anemia, the issue is that when their red blood cells let go of oxygen, it twists and distorts the hemoglobin molecule so that it can never pick up oxygen again. And through that twisting and distorting, it makes that red blood cell lose its nice donut shape. And instead of just being folded over or kind of smushed a little bit, it turns into a sickle. So that red blood cell is no good anymore. So people with sickle cell anemia have a host of problems, one of which is low oxygen carrying capacity of their red blood cells. So if we can turn adult hemoglobin into fetal hemoglobin for people that have sickle cell anemia, then that means that those red blood cells that they do have and have not turned into that sickle shape will have a good affinity for oxygen and be able to drop and carry more oxygen than they normally would. So here what we're looking at are some sickled red blood cells, and remember, let's kind of take a little blast from the past, if you will, a few minutes ago when we talked about the coloration of red blood cells. If we have this nice bright red color, who is attached to that iron? Oxygen, absolutely right. When we have this darker color, do we still have oxygen attached? Nope, no oxygen attached to it. And remember what I just said about sickle cell anemia. Notice these kind of malformed red blood cells, like this is a beautiful sickle shape, just absolutely stereotypical sickle shape. It's not like the nice flattened donuts that we wanted to look at. So with sickle cell anemia, the problem is not picking up oxygen, it's when they let it go, how it distorts and it malconfigures the hemoglobin molecule, which in turn malfigures the red blood cell. So these red blood cells have dropped off their oxygen. You know that because they're a darker color now, but also because they now have that sickled shape because their hemoglobin molecule has become distorted. So low oxygen carrying capacity is an issue with sickle cell anemia, blood clotting in various places because these cells don't travel through capillaries very well. They don't form that rouleau that we talked about, that's a nice little stack of dinner plates. They get stuck in different places. So as a result of those clottings and the inability of the red blood cells to travel nicely through joints and small areas, it can cause a lot of pain. People that have sickle cell anemia routinely will have to be on lots of different medications and have blood transfusions. Hemoglobin conversion and recycling. All right, so far what we have talked about is mostly anatomy type stuff. We've talked about what's in whole blood, what's in the formed elements, the job of the red blood cells in the formed elements. We haven't really talked a lot about anything else, the platelets or the white blood cells. We'll get to those soon. And we've also talked about those plasma proteins, what their job are. We talked about several different things that were more anatomical in structure. This is the first chance that we're going to get to actually start talking about some physiology. And if you've had me for A&P1, then you probably already know that if it's a process, if it's a physiological process, or how something does it, not what it does, but how it does, those are good candidates for short answer questions. So I would star or asterisk the next couple of slides where we talk about how hemoglobin is converted and how it is recycled. So keeping in mind that red blood cells only live about 120 days or so. So when it's time for those red blood cells to die, or when they go through apoptosis, or they're no longer useful after that 120 days, we don't want to throw out everything. We don't want to throw out the baby with the bathwater. There are some things that we can actually save. So the phagocytes are going to break down, and phagocytes are just any of your cells. They could be monocytes, any large eating cell that can eat stuff. Phagocytes break down hemoglobin into its components. And remember, hemoglobin has those 4 chains, 2 alpha and 2 beta. The alpha and the beta chains are then going to be filtered out by the kidneys and the urine. So the other parts that were broken down by the phagocyte that it doesn't really need, those will go out of your body and those will be considered as waste. However, a nice healthy set of kidneys, they're going to be, the alpha and beta chains are filtered out. Hematuria happens after kidney damage, or there's been damage to the vessels along the urinary tract, which means that you're having red blood cells in your urine. Globular proteins can become amino acids. So those globular proteins, or those alpha and those beta chains that we saved, we can break them down, because the alpha and the beta chains that make up hemoglobin, and hemoglobin is a protein. Proteins are made out of amino acids. So we're kind of going back. We're deconstructing this, kind of chopping down a car. So we've gone from the hemoglobin to break it down into the alpha and beta chains. We keep those in circulation, and then we break down those alpha and beta chains into their individual amino acids. Now we have the issue of dealing with that heme that has that iron that's attached to it. Well, heme, we want to be able to use as much of it as we can. So heme is then going to be converted into biliverdin once we take the iron off of it. Iron is then going to be transferred with a molecule called transfurin. And we'll finish this discussion up in just a second. So, hemoglobinuria means that the hemoglobin breakdown products in the urine, because you have excessive hemolysis in the bloodstream. You've had way too many red blood cells explode and break for some reason or another. That is not a normal thing that needs to happen. So what we find is that in the urine we'll have a lot of hemoglobin, which it shouldn't be. Hematuria is where you have whole red blood cells in the urine as a result of the kidney's inability to filter out those red blood cells, or there's been some tissue damage. Now, continuing with where we were going. So once again, to recap, we had our red blood cells. It's broken down, no longer usable after 120 days. We don't want to throw out that hemoglobin. So we have phagocytes that will break it down into the alpha and the beta chains. Alpha and beta chains will stay in circulation after they've been filtered out by the kidneys. Those alpha and beta chains need to be further broken down into the individual amino acids that make up those alpha and beta chains. Now we have to deal with that heme. That heme group is going to be converted to biliverdin once we remove the iron from it. Biliverdin, if you've ever taken a Latin or a French or a Spanish-speaking class, verde means green. So biliverdin is green. It will be eventually converted to bilirubin. Bilirubin you're familiar with because it is a yellowish sort of color to it. Bilirubin is excreted by the liver or in the form of bile. Jaundice is caused by a buildup of bilirubin because you have liver damage, or if you're a newborn, you have a brand new liver, never really used it before, so you're just kind of working the kinks out. So it's why sometimes newborn babies will have a touch of jaundice. It does not mean they have liver failure or damage. It can sometimes, but usually it doesn't. It just means the liver's hasn't really worked itself out yet. Or you have some sort of bile ducts for adults. An adult with jaundice, you screwed up your liver somewhere along the way, whether it was congestive or it was from abuse of drugs and alcohol or something. Something happens with your liver. That's when you have that jaundice. This bilirubin, as it pertains to the recycling of hemoglobin and the breakdown of red blood cells, is converted by intestinal bacteria to urobilins and stercobilins. These two items are as a result of from biliverdin to bilirubin to urobilins and stercobilins. We only get to these two items here with the intestinal bacteria that live in your gut, like E. coli. So now the iron. What happened with that iron? We stripped the Hemi group of that iron there, and then we no longer called it Hemi, got converted into biliverdin and then bilirubin, and then by the intestinal bacteria to urobillions and stercobillions, whatever happens to the doggone iron. Well, that iron is transported by transferins, which are special transport proteins, and then they're eventually transferrin will transfer them to ferritin and hemolystrin, which are storage proteins. So we transport and then store this iron so that we can use it again when we need to make more red blood cells. And here is a beautiful picture of that entire story that we just went through. So we're going to go over it again in just a second, but I want you to take a moment to see if you can kind of digest what's happening in this picture. So we want to start here. So start here and follow your way along and see if this kind of starts to make sense to you a little bit. So just let it digest for a second. All right, so that macrophage that we talked about, that phagocytotic cell in the spleen or the liver or the bone marrow, it takes that red blood cell that's been hanging out. So we have the red blood cell that's been doing its thing. After 120 days, 90% of those red blood cells are going to be phagocytized. Now, some of them may rupture in the bloodstream, and then just the fragments get phagocytized. So 90% of it, the whole red blood cell, it's no longer doing its job, it's no longer needed in the body, it's old, it's defunct now, it's time for the new crop to come in. 90% of them are just going to be phagocytized. A small percentage of them, they just don't get a chance to be phagocytized in time, so they just lyse or burst, or what we call hemolysis, in the bloodstream. And then even some of those, a good chunk of those, actually 10% of those, the other 10% will be phagocytized, those fragments. Now, if we do not get a hold of this 10% of red blood cells on average that just lyse or burst in the bloodstream, hemoglobin that is not broken down and the alpha and beta chains will be eliminated in the urea. So that was the hemoglobin urea that we talked about. See how it's going through the kidneys and it's being eliminated out? We didn't want that to happen. So this right here is something that can happen, but it really shouldn't be happening because we want to recycle that hemoglobin. And if it does happen in most healthy people, it's very minimal and you don't even notice the issue. If you have problems happening in your body, if there's something that's not quite right, then you'll see a higher concentration of that, and your doctor will be able to measure that, and it will be other tests will be administered to figure out what's going on. All right, so now that we've phagocytized our red blood cell, we took those alpha and beta chains, broke them down, removed the hemi from them, broke them down into the amino acids, and these amino acids, notice where they're going. They're going back to the red bone marrow because that is the site of red blood cell formation. And you learned that in AMP1 in the bone discussion, in the bone chapter. That's a part of the red bone marrow is that it makes red blood cells. So now that we've taken care of the alpha and the beta chains, broken them down into amino acids and then transferred them back into the red bone marrow, now we've got to deal with the hemi group. With that hemi, we remove the iron and it's transferred by transferrin, and then it's stored in the red bone marrow as well. So now we have two things that are absolutely required to make a red blood cell in the red bone marrow. We have iron, that's going to be the center of the hemoglobin, and then we have the alpha and beta chains that will link up together, or the amino acids that make up the alpha and beta chains that will be linked together and turn it into hemoglobin. So we have to get rid of this hemi. It's no longer a hemi anymore. It's called biliverdin, and then it gets converted to bilirubin. Bilirubin will bind to that plasma protein that we talked about. 60% of your plasma proteins are these transporting plasma proteins, albumin. So it transports it through the bloodstream into the liver, and then the liver will then excrete out via the bile, and this is a bile duct, and it's kind of making its way into the large intestines. It actually goes into the small intestines first, and then makes its way into the large intestine. Well, be more accurate, it goes to the pancreas, and then the large intestine, but I digress. Eventually, it goes to that bile duct, that bilirubin, then because of the activity of the intestinal bacteria, turn bilirubin into urobilin and stercobilins that we eliminate out in the waste, or some of them can be absorbed into the circulation and eliminated out in the urine. This is definitely going to be an option for a short answer question if I ask you how to recycle the components of a red blood cell after its 120 days stay in the body. So make sure that you're very familiar with this process. If you want to draw it, that's fine. Just make sure that you explain to me what you drew. You want to make bulleted lists? I'm cool with that as well. You just want to write paragraphs. That works. Just show me that you understand what's going on. Now, on the other side of this, how do we make red blood cells? We talked about how we can break them down, but how do we actually make them? Well, red blood cell production is going to be directed under the hormone of erythropoietin. The process of making red blood cells is what we call erythropoiesis. Erythropoiesis, that process of making red blood cells, happens in the red bone marrow, or what we can also call the myeloid tissue in adults. We do have adult stem cells that mature to become red blood cells. And where do you think those adult stem cells are found? In the red bone marrow. Hemocytoblasts are the name of those adult stem cells that are found in the red bone marrow. They divide into two camps. So we've looked at how to recycle red blood cells. We haven't talked about how to make red blood cells. When it's time to make red blood cells, we're going to not only make red blood cells, we're also going to make white blood cells, and we're going to make platelets. So we're going to make all of the four elements, because that stem cell, called a hemocytoblast, is going to divide into two camps. The two camps that it divides into are what we call myeloid stem cells and lymphoid stem cells. Myeloid stem cells are going to be the ones that are responsible for making the red blood cells, which we were initially talking about, but it's also going to make 4 out of those 5 classes of white blood cells. And as you'll remember from lab, we have 5 classes of white blood cells: neutrophils, eosinophils, basophils. Those are all your granulocytes. And then we have monocytes, and then we have lymphocytes. The 4, eosinophils, neutrophils, basophils, and monocytes, are made from myeloid stem cells. The lymphoid stem cells only make one type, and that's a lymphocyte. This discussion of what we're talking about now, the process of making red blood cells, or erythropoiesis, another excellent option for a short answer question, so I would start asterisking these slides as well. So hemocytoblast, that adult stem cell that's found in your red blood marrow, divides into 2 camps: myeloid stem cells, lymphoid stem cells. And the myeloid stem cells make your red blood cells and 4 out of 5 white blood cells. And the lymphoid stem cells and myeloid stem cells also make the platelets. They come from that camp too. Lymphoid stem cells only make lymphocytes. So to get this process going, there's a couple of things that we need. We need amino acids. Did we not show how amino acids can be found or will be found in the red bone marrow after we have recycled an old red blood cell? Check. We need iron. Did we not discuss how iron is going to be transferred and stored to the red blood cells or the red bone marrow after an old red blood cell has been broken down? Check. We also need vitamin B, vitamin B12 in particular. Well, we didn't really get that from the red blood cell that we broke down, but we do get that out of our diet. So from dairy, if you're a vegetarian, and meat products, vitamin B6 and folic acid, those are things that we get from our diet. So we can get the vitamins and the folic acid that we need to get this party started from our diet. Check. Now, people that have what we call pernicious anemia, they have a low red blood cell production, and the reason their red blood cell production is so low is because of their diet. It's not necessarily because of there's a hormonal issue. That just means that they're not getting enough vitamin B12 in their diet. We see this sometimes for people that are on very strict vegan diets, people that are starving, and for patients that have had a gastric bypass, if they're not taking their vitamin B12 shots because vitamin B12 absorbs through your stomach, and after you've had gastric bypass, it reduces the surface area of your stomach, so you're not absorbing as much of that B12 from your diet, so they have to give you a vitamin B12 shot. Not only do we need amino acids, vitamins, and iron, we also need hormones to stimulate this whole process, to get this party started. The hormone that we need is erythropoietin. Erythropoietin is a glycoprotein that's made in your kidneys and your liver. We talked about erythropoietin at the end of ANP1. I keep saying that we talked about this before, we talked about that before, just to kind of jog your memory about these things. So you can sometimes call EPO erythropoietin stimulating hormone, but we're just going to call it erythropoietin. Erythropoietin will be secreted from your kidneys when the tissues are hypoxic, which means that the oxygen level is low. So what that triggers and tells the body is that the oxygen levels are lower than we need them to be, so we're going to have to make some more red blood cells. We need some more oxygen carrying capacitors. We can have this hormone that's made when you're in higher altitude. You'll be stimulated to secrete more of this hormone when you're at a high altitude, or some forms of disease that cause the tissue to be hypoxic will also stimulate this hormone to be released more. And the reason high altitudes will cause you to release more erythropoietin is because there's not as much available oxygen at higher altitudes. And which is one reason why athletes will do what's called high altitude training. We know that red blood cells carry oxygen. Check. The more red blood cells you have, the more oxygen your body will have. Check. We know that oxygen is responsible, is one of the main and best, most efficient ways to make energy, or ATP. Check. If I'm an athlete that's going to have to run a marathon, or I'm going to have to beat the crap out of somebody in a UFC fight or a boxing match, or if I'm a football player, or if I'm a swimmer, or whatever it may be, if I want to do that process for longer, I need more ATP. To get more ATP, I'm going to have to get some more oxygen. To get my body to carry more oxygen, I need to make more red blood cells. If I need my body to make more red blood cells, I can, what we call blood doping. I can get a shot of erythropoietin, or EPO, and make my body make more red blood cells, which is usually frowned upon in most professional athletics, because that's what I think Lance Armstrong got popped for. Or I can high altitude train. So if I high altitude train, then my body will be stimulated to make more red blood cells, so when I come back down to my normal level, then I have a higher concentration of red blood cells and more oxygen carry capacity and more energy to do my event. But keeping in mind that when I do that, just high altitude train, that it doesn't give me as long a lasting effects as if I just have my doctor give me a shot of EPO. That's much easier to do, and you have more energy. And I think another thing that Lance Armstrong did was that he would take his own blood and store it, high altitude train, and then right before his race, he'd have his own blood pumped back into him, so he's got more red blood cells. So that was part of what he did, and it was clearly frowned upon by the cycling community. So the things that we just talked about as far as making red blood cells and recycling old red blood cells, great questions for short answer questions. So for this blood typing business, I'm not going to spend any time on this at all. I'll let you guys just read over this and let this digest. If you have questions over it, first view our virtual lab, and then if you still have questions, then please email me, come see me, so that we could get those questions straightened out for you. All right, so we've already had this discussion, so that's why I'm not going to talk about it any further, because there's a recording that's on it. So the other class of formed elements 4 of which that are formed from those myeloid stem cells are your leukocytes, your white blood cells. They don't have any hemoglobin. Unlike red blood cells, they do have nuclei, they do have mitochondria, Golgi apparatus, or those other organelles. Basic jobs of the white blood cells is that they are meant to have an immune response, so defense against pathogens. They will also help remove toxin and waste, and they'll attack any of your own cells that are abnormal. We're just going to give an introduction into who are white blood cells and where do they come from. How they do what they do, we'll talk about that in the lymphatic system. 4 characteristics of white blood cells is that they can come in and out of circulation, so they can migrate in and out of the bloodstream. They have an amoeboid-like movement, so they kind of look like amoebas with little pseudopods that come out and crawl. They are attracted to chemical stimuli, and they'll go towards something like chemicals that are released from platelets during the clotting process. They will migrate towards that, or to red blood cells that are dying. They will migrate to those. The macrophages or the monocytes can do that to break them down, and we call that positive chemotaxis. The 3 that are considered to be phagocytotic that can eat other cells are neutrophils, eosinophils, and monocytes. Basophils and lymphocytes don't really have that phagocytotic activity that can take place with them. So for these 5 different classes of white blood cells, remember we said that 4 out of those 5 classes comes from the same stem cell, the same myeloid stem cell that red blood cells come from. And look at what we notice here. Neutrophils, eosinophils, basophils, and monocytes are the same 4 white blood cells that I said arise from myeloid stem cells. They all have a role in your non-specific defense, which means they don't really have a specialized way that they treat pathogens. They kind of just treat all pathogens the same and try to get rid of them. The one white blood cell that comes from the lymphoid stem cells, it does have a specific defense, and those are lymphocytes. So it makes sense that lymphocytes have their very own stem cell that they arise from because they have a much more special role. Lymphocytes can then, they treat different pathogens differently. So it will treat an infection with E. coli different than it would treat an infection with Staph aureus or would treat an infection with H1N1. They're more specialized and they're more specific. So they're highly specialized cells. So how neutrophils work, and so we'll look at these next couple of slides here how each of these white blood cells does their job. 50 to 70% of your white blood cells are neutrophils. They are phagocytotic. They do have the granules. Degranulation removes the granules from the cytoplasm. We have defensins as part of those granules that will attack pathogen membranes, and it can also release chemicals, prostaglandins and leukotrienes, which are chemical messengers that will attract other white blood cells to the area. When we have this neutrophil activity, we can also have some pus that's formed. So typically, what you find in your pus are the remnants of neutrophils that have done their job in the bacteria that they have decomposed. Eosinophils are also called acidophils. 2 to 4% of your circulating white blood cells are eosinophils. They attack large parasites, and they're able to do so by releasing their granules, which are things like nitric oxide and cytotoxic enzymes. Eosinophils are very sensitive to allergens. They can elicit an allergic response. They help to control inflammation with enzymes and elicit inflammatory effects of the neutrophils and the mast cells. So when the neutrophils and the mast cells, specifically the neutrophils, are releasing their chemical messengers, the eosinophils can release their own special chemical to help counterbalance that chemical messenger so you don't have too much inflammation. Inflammation sounds the alarm, and it says, hey, there's a problem in this area, but too much inflammation can be a problem. And so to help balance that out in regards to neutrophils, eosinophils release chemicals that will do that. Basophils, less than 1% of your white blood cells are basophils. They have a tendency to accumulate and damage tissues. The granules that are in basophils that are released are histamine, which helps to dilate blood vessels, and we'll talk about this discussion of histamine and the dilation of blood vessels for quite a bit throughout the next couple of chapters. Dilation of a blood vessel means that we make it larger. And then they also release heparin, and heparin's job is to dissolve blood clotting, which is also something that we're going to talk about at great length towards the end of this chapter. We talk about the blood clotting process. Monocytes are now we're in the class of agranulocytes. They don't have those granules on there. Remember, those are those really great big giant white blood cells that we saw that had kind of that kidney bean shaped nucleus, sort of nucleus of this is my giant cell. We had a nucleus that was kind of shaped like that, sort of. And then for our neutrophils, they had that multi-lobe nucleus that was kind of shaped like that. So that would be a neutrophil. This would be a monocyte. And then for the eosinophils, they had the bilobe nucleus. And then something like that. And then for the basophils, they just look like, like a lot of stuff going on in there. Nucleus is just like all over. So that was the eosinophil. This one here is a basophil. And they all, the basophil, the eosinophil, they had little granules in them, and so did the neutrophil. But the monocyte, the one we're talking about now, did not have those granules. Large, spherical. If a monocyte gets out of circulation, we call it a macrophage, engulfs large particles and pathogens, including red blood cells that have life or burst or they're getting ready to die. And they will secrete substances in much the same way that neutrophils to attract other immune cells to an area, and even to attract fibrocytes to an area to repair that injury. And then our last type of white blood cell was, we had our lymphocytes. Remember, lymphocytes weren't that much bigger than a red blood cell, about 20 to 30%. The second highest concentration of white blood cells are your lymphocytes, and they had a great big giant nucleus. So their nucleus kind of took up almost the entire cell. They also, like the monocytes, will migrate in and out of the blood. We find most of them in your connective tissues and in your lymphoid organs, such as your spleen and in your lymph nodes. And they have a big role, big important job. They are the only of your white blood cells that have a role in your specific defense system. And remember, specific defense for right now, because we haven't gotten to that part yet, it just means that the way that it attacks a pathogen is going to be different depending on what that pathogen is, because lymphocytes can differentiate or divide into alpha cell, or B cells and T cells. The three classes of lymphocytes that we have, differentiating are T cells, which elicit cell-mediated immunity, B cells, which give you antibody-mediated immunity, and then we have natural killer cells, which don't really have a specific role. They treat all pathogens kind of the same way, but they detect and destroy any abnormal tissue cells. So, like cancer, any of your cells that become cancerous, natural killer cells will notice that their cell surface marker isn't what it should be and will target it. The two that are responsible for your specific defense would be your T cells and your B cells. And once again, we'll talk about this at length in lymphatic tissues. So performing a differential count is where we look to see what is the concentration of each one of these different classes of white blood cells, one of those 5 classes, and we see if that number is in its normal range. So if it is if we had a neutrophil, is it within that range of 60%? If it's a lymphocyte, is it in the range of 30%? If it's a basophil, is it in the range of 1%? So we've looked to see, and remember we talked about this in lab, that you'll count 100 white blood cells and then tell me how many of those 100 white blood cells fit in each one of those categories of white blood cells, each one of those 5 categories. So white blood cell disorders that we can find are leukopenia, which means that you have an abnormally low white blood cell count. leukocytosis, which means you have an abnormally high white blood cell count. If you have a leukocytosis, that's usually indicative of your body trying to fight some infections. You've got lots of white blood cells. They have become they have proliferated, lots of them, made lots of them. That usually means that something's not quite right. Your body might be fighting an infection. Leukopenia, on the other hand, that might tell us that your body's not making enough white blood cells, and we don't know exactly why. That could be an issue with your stem cells, your myeloid stem cells. It could be an issue with the bacteria overrunning your body, and it's killing those white blood cells before they have a chance to really mount up their defenses. Leukemia is the far end of leukocytosis. That's an extremely high white blood cell count. And if you have a white blood cell count that's high enough to be characterized as leukemia, chances are that the white blood cells that keep making copies of themselves and don't listen to the stop signals, they ignore those stop signals, are cancerous. Cancerous cells just do their own thing. They say, screw whatever the body tells me to do, screw whatever I'm supposed to be doing, I'm going to do what I want to do, and I want to divide, and I'm going to make lots of myself. How white blood cells develop is not all that different from the way that red blood cells develop. They don't need iron. They don't need the same kinds of amino acids from those, to make that globular protein of hemoglobin. But the place where they develop primarily is in the bone marrow, except for the monocytes. Monocytes develop into macrophages in the peripheral tissues, and then they're fully mature once they are free macrophages in peripheral tissue. So monocytes can develop outside of the red bone marrow. Everybody else develops kind of in the red bone marrow for the most part. To make these different kinds of white blood cells, even though they come from the same myeloid stem cell, there is some differentiation or differences that have to take place because neutrophils have their job, eosinophils, monocytes, basophils, they all have slightly different jobs. So there are different hormones that will stimulate their production. So the M colony stimulating factor will stimulate monocytes. G colony stimulating factor will stimulate any of those granulocytes, so neutrophils, eosinophils, or basophils. The G and M colony stimulating factors will stimulate the granulocytes, those three that we just mentioned, as well as the monocytes. And then the multi-colony stimulating factor is going to accelerate the production of the granulocytes, the monocytes, the platelets, as well as the red blood cells. So far, we have only really talked about one hormone being responsible for the formation of formed elements, and that was erythropoietin to make red blood cells. Now what we're looking at are these other hormones or factors that will differentiate and cause for these various different types of white blood cells to be made. You'll notice on this list who's missing is the lymphocyte stimulating hormone. And that's because the lymphocyte stimulating hormone is only going to be regulated to the lymphoid stem cells. So here we have that whole process of formed element formation. In the red bone marrow, we have our hemocytoblasts that will differentiate, and keep in mind this is all under hormonal control. A lot of the other hormones that are responsible for this we just haven't talked about, and we just don't have the time to talk about all of those. We have our myeloid stem cells, we have our lymphoid stem cells. We'll start with lymphoid stem cells because they're the easiest. They become only lymphocytes, they become lymphoblasts, and then pro-lymphoblast, and then finally a lymphoblast. And then we have our myeloid stem cell, which just kind of hangs out and does its thing until it's under the direction of multi-colony stimulating factor. And remember from the previous slide, it means it can make red blood cells as well as platelets as well as white blood cells. So we're going to differentiate into those three camps. We have our red blood cell precursor, we have our platelet precursor, and we have our white blood cell precursor. Let's go ahead and work with our white blood cell precursor. If it is under the hormone of G colony stimulating factor, that myeloblast is going to differentiate into those different types of granulocytes of the basophils, the eosinophils, and the neutrophils. They first become myelocytes, and then they become band cells, and then they finally differentiate into those actual basophils, eosinophils, and neutrophils. We can have this other cell. It kind of divides into two camps, those that make the granulocytes as a myoblast, and then we can have a monoblast over here that when it's under the control or direction of monocyte colony stimulating factor, then it becomes a promonocyte, and then finally a monocyte. And for our platelets, notice that we don't really have a factor that's responsible for making the platelets. It's just kind of a myeloid stem cell that gets really, really big and then bursts into these little tiny fragments, and we call those little tiny fragments platelets. And then for our red blood cell precursor, still came from that myeloid stem cell that came from that hemocytoblast. When it's under direction of erythropoietin, it becomes a pro-erythroblast. That erythroblast has stages, and one of the stages in there is the ejection of the nucleus. Once the nucleus comes out, it's called a reticulocyte, and after a little bit more maturation, it becomes an erythrocyte. So this slide you should be very familiar with. You don't have to replicate it and draw it again. You can if you want to, but you should understand it to be able to answer questions off of it. You should be able to explain where lymphoid stem cells come from, where where monocytes come from, where eosinophils, basophils, any of those granulocytes, and where erythrocytes come from, and platelets. You should know the role of those different colony stimulating factors. What does multi-colony stimulating factor do? Granulocyte, monocyte colony stimulating factor, monocyte colony stimulating factor. What is the role of erythropoietin? What do we call a red blood cell that has lost its nucleus? A reticulocyte. What do we call a cell that is just derived from a lymphoid stem cell? A lymphoblast. So you should know this chart here. You should be very familiar and very comfortable with it, because this whole discussion is fodder for, it's grandfather for a short answer question on your exam, and also for multiple choice questions. So I think that's where we're going to wrap it up for today. We will start off with a discussion of platelets, and we'll talk about clotting on the next session. All right, have a great day. Enjoy the weather, enjoy the day, enjoy your lives, and I will see you either physically or virtually very soon. Bye.